This lecture includes a comparison of passive and active transport, and then has details about active transport and bulk transport. So here's our little chart. We've kind of talked about it. So passive transport goes from high concentration to low, the natural tendency, versus active, which goes from low to high, shoving into an area that's already concentrated. That means, I think of it as a triangle. If we're going from high concentration to low, we're going down the concentration gradient. And if we're going from low concentration to high, we're going against the concentration gradient. The concentration gradient just means the difference in concentration across a membrane. Energy requirement. No additional energy required for passive. Additional energy required for active, frequently in the form of ATP, but not always. Then we come to transport proteins. So passive transport, the two types, simple diffusion does not use transport proteins, but facilitated diffusion does. So we could say sometimes passive transport uses them. To pump against the gradient, you'll always need a transport protein for active transport. What types of proteins? So channel proteins and carrier proteins can be involved in passive transport, facilitated diffusion. In active transport, it's only carriers, which we sometimes call pumps. It can't be channels, because channels are just corridors through which solutes pass, so this would be the channel, and so that can't go against the gradient because it's just flowing. Subcategories, we said simple, so that would be things like the gases. We said facilitated. That would be, some examples of that are water movement, so aquaporins. We have glucose transport. And those are some amino acid transport from high to low. And then examples of active, a lot of times we talk about the Sodium-Potassium Pump. It's important for neurons. We talk about hydrogen ion or proton pumps. Those are going to be important for photosynthesis and cell respiration. And finally, what about solutes that are moving against their concentration gradient from low to high? That's going to be active transport. We've already seen the basics of that. Pump is important for building up concentration gradients so that your nerves are able to signal. So this is how a nerve works. Here's the sodium-potassium pump. Lots and lots of sodiums outside, lots of potassiums getting pumped inside. Then you have channels that open at different times in nerve firing, and when the channels open, some of them are potassium channels, so potassium flows back outside the cell. Some of them are sodium channels, so sodium flows into the cell. And this basically changes the charges across the membrane because it moves the ions, and that makes sure the next one open up, and the next one open up, and the next one open up, and it basically moves your signal down the nerve. Pumps that move ions are called electrogenic. So basically it's because they create a membrane potential, which is when you have positives and negatives in different amounts on the sides of the membranes. So the chemical force is the ion itself's concentration gradient. So where is sodium high, where is sodium low. Electrical force is the plus minus, and where's that pluses and minuses distributed across the membrane. Pumps in general are quite useful for creating concentration gradients across the membrane. So they'll pump, in this case, the ions to one side in large amount over the other. And so you'll have unequal amounts of ions. And so basically it's like water behind a dam, that kind of analogy, where the water is poised to come out of the dam to do work. And so the same thing here, you poise the ions in one side or the other of the membrane. You can also do this with other molecules, but active transport's what's used to get them there and make this steep concentration gradient so it can do work later in the future. So why does that matter? So here's a sodium-potassium pump, and here is a sodium channel and a potassium channel. This is how nerve signaling is going to work. So the pump is going to pump the sodium out to where it's already high and potassium in to where it's already high. You'll notice that you put 3 sodiums out for every 2 potassiums in, which means overall you're getting 3 pluses outside and 2 pluses inside, which overall makes the outside of the cell positively charged and the inside of the membrane of the cell negatively charged. So how's that going to influence sodium and potassium when they try to move back across the membrane? So let's look at this. Diffusion of sodium ions here is facilitated by or impeded by the sodium concentration gradient. So we have to look. Okay, so sodium is going to be moving this way. Is it promoted or impeded? So we look going from high to low. So that would be that it is facilitated or helped by the concentration gradient. Now let's look at the potassium. Potassium moves in this direction. So is that facilitated or impeded by the potassium concentration gradient? It's going high to low potassium, so that one is also facilitated. So the chemical force in this scenario helps move both the sodium and potassium ions passively across the membranes. So what about when we look at the electrical forces at work here? All right, so let's look at our sodium ions again, and we're going to look at charges. So the sodium ions are positive. They want to come inside. So inside's negative. We know that opposites attract, and so that's going to again be facilitated. So what about potassium? Potassium is positive. Potassium wants to go out. And so look at this though. Potassium's positive going towards the positive. So that means that potassium is impeded by the electrical gradient. So overall, both of them have a facilitating chemical gradient in their respective directions. But sodium also has a facilitative electrical gradient, whereas potassium has an impeding one. So overall, the sodium has an electrochemical gradient that's larger than the potassium because both the chemical and the electrical add together. So that just means that sodium is going to flow into the cell more easily than potassium flows out of the cell. And these are both passive transport channels. The active transport just continues to pump in the direction that keeps building these electrochemical gradients. Proton pumps are also electrogenic because they are pumping ions, so the positive charge is going to build up, in this case on the outside, in the extracellular fluid, and in this case the negative is on the inside. This is an active transport, so it does take energy, in this case in the form of ATP. So this is called co-transport. So co-transport is a form of active transport, and that is because it uses energy and moves solutes against their concentration gradients. So here is energy usage, and it's pumping hydrogen ions outside the cell, protons. Those protons then diffuse back into the cell. That part's passive transport. But as they diffuse into the cell, in this case, they can take sucrose along with them. Sucrose is going from low sucrose to high sucrose, making this part active transport, low to high. So we have active transport because of the ATP for the pump. We have active transport because we're putting the molecule that comes along for the ride for the co-transport. That's going from low to high. So it doesn't have to go both in the same direction either. Some co-transporters, one goes in and the other one comes out. Both of those are still the same. Basically, it's relying on a concentration gradient, and as that flows back down its concentration gradient, it will do the active transport of the other substance. So here's an example of a sucrose hydrogen ion or sucrose proton co-transporter. So this would be one way you could get sugars into cells that are already full of sugar. Bulk transport is the last type of transport, and basically this is for moving large molecules across the membrane or taking large quantities of small molecules in or out at one time. So this is when cells are going to secrete molecules or do things like ingesting molecules from the environment. Well, there's two different types, exocytosis and endocytosis. So exo, like exoskeleton in an arthropod, exo means outside. So exocytosis is dumping products outside the cell. So here's your cell, and here's your little vesicle full with products. the vesicle is going to fuse the plasma membrane, and then, like this, it will dump those products out of the cell. That's exocytosis. Endo means in or inward, so endocytosis is the opposite. So here's your cell. Here are some products that are going to get taken in. The cell makes a little pocket. We'll mark the membrane there that surrounds those particles, and then those are going to get pinched in into a little vesicle and taken into the cell. So that's endocytosis. So what types of cells do lots of endo and exocytosis? So exocytosis is going to be done by secreting cells, particularly those secreting proteins. So these vesicles are basically the ones that come from the Golgi, from the ER prior to that. Those are the ones frequently that are the ones that are undergoing exocytosis. So good examples of eating cells are amoebas and white blood cells. Both of them are going to intake particles in order to break them down inside. The amoeba will break it down for food purposes, and then the white blood cell will break down the virus or bacterium or other invader to protect the body. One type of endocytosis is called phagocytosis or phagocytosis. So this is the one that uses these pseudopodia in order to capture a particle from the environment, in this case food. This is like an amoeba surrounding its food. Then once it gets surrounding it, then the phospholipid bilayer rearranges itself and basically fuses with itself, and the inside part here will become its own little vesicle. And this is of course not open, this is a cut through. This is actually a sphere containing the food inside. So then what might happen when this comes inside with food? Well, that could fuse with the lysosome, and the lysosome hydrolytic enzymes might digest that food. Then the food can get shipped other places in order to be used either in cellular respiration or for building new cell parts. Pinocytosis is taking whatever solute is outside, taking it in the inside of the cell in relatively small amounts. So this would be, instead of having these extensions, the cell membrane just kind of folds inward and pinches off into these little vesicles that are going to take whatever's out there inside. Receptor-mediated endocytosis is similar to pinocytosis in the fact that it just pinches in, but in this case it has receptors that are specific for a particular substance, and so here are ones that are specific for triangles. So when you have receptor-mediated, you're going to get a lot more triangles coming in than in pinocytosis when you just get a random assortment based off of what's in the fluid. So receptor-mediated endocytosis is selective. So the LDL, low-density lipoprotein receptors, are on cells that help bind the cholesterol to take it out of the bloodstream and then ship it into, get processed into the liver. So that LDLs, those are going to be useful for removing cholesterol from the bloodstream. And different people have different amounts of LDLs, which can change how much cholesterol they can take out of the bloodstream and significantly impact people's health.